biology class. Today we are going to study about chapter 11, Biotechnology, Principle and Processes. Before we start the chapter, we should understand what is biotechnology. Biotechnology in a layman's language or in a very simple language is utilizing the organisms so that they can be used for the benefit of human mankind. We are going to do certain genetic manipulation, we are going to change them so that they could best fit the human mankind. So, by this definition, even the formation of curd in which we are using the living organism, that is lactobacillus, to convert the milk into curd is biotechnology, or brewing of wine where we are using various varieties of Saccharomyces cerevisiae or yeast is also biotechnology, or even the leavening of the bread where we are using yeast to make bread is biotechnology. On the other terms, very specifically the term biotechnology is used to make in vitro fertilization, that is generation of test tube babies, to make transgenic plants and animals which we call as genetically modified organisms or GMOs. To cut down the controversies or to make the definitions more clear, according to European Federation of Biotechnology, the definition of biotechnology is the integration, means the combination of natural science, what all natural processes are taking place, and the organism. Either it could be a complete living organism, a microbe, a plant, or an animal, or its cells, and the parts thereof, that it could be its DNA or RNA or protein or any of the signaling molecules and molecular analogues of the products and the services. So integration of all of them together is biotechnology. Two core methods that give birth to biotechnology is genetic engineering and maintaining the sterile conditions. Now, what is genetic engineering? The term engineering means that you are changing or manipulating something to create something better. Similarly, genetic engineering means to manipulate the DNA or RNA, whatever is the genetic composition, so as to get a better type of living organism. For example, there is a plant who is very much susceptible to a pest. By doing some alteration in its DNA, we are going to create a plant which is now resistant to that pest. That is genetic engineering. Next is maintaining the sterile conditions. For example, for making antibiotic, I want only one type of microorganism to be present in my preparation. This is a medium. I want only one type of bacterium to grow in that particular preparation. I do not want any other microbe to be present here. Although the microbes are present everywhere, they are omnipresent, I want the condition so that only one type of microbe is present in a particular bioprocess. Under such a situation, I need to create a particular condition where the entry of all other microbes is restricted and only whatever type of microbe I am adding is present. That means this condition is a sterile condition. That means in the air there is no microbe, or all the surfaces or the person who is working with this particular bioprocess is free of all the microbes and only the desired microorganism is free. This is known as sterile condition. So maintaining such a sterile condition is another major reason for the giving birth to biotechnology. You must have studied in your lower classes two types of reproduction, the sexual reproduction and asexual reproduction. In the sexual reproduction, the egg and the sperm with different genetic composition, they fuse together to form the zygote. So zygote is having more of variation because it is getting genetic information from two different types of individuals in the form of the egg and sperm which fuse together to form it. However, in asexual reproduction, in the same particular parent cell, the DNA is getting duplicated and the cells are then dividing together to give rise to two daughter cells. So genetic variation is more there in the sexual reproduction, and this principle is being used in various of traditional methods which we are using in animal breeding and in agriculture, where we have tried to fuse the sperm and the egg of the high yielding, high milching cattle or the resistant cattle to get the breeds of the animals which are best fitted to our requirement. This is the advantage of sexual reproduction, where you can add on the varieties, you can get the desired features as per your convenience. However, in asexual reproduction, this is not feasible. So why we are doing this type of manipulation traditionally? So that we can add on those genes which are required, which are more beneficial to us, and dilute of those genes which are not required, which are not beneficial for the human mankind. Now in biotechnology, all this purpose can be fetched by three basic techniques, that is recombinant DNA technology, gene cloning and gene transfer. In recombinant DNA technology, we are going to do the combination of the genes which are best fitted, and we are going to introduce them into the host. In gene cloning, we are going to get the best variety, best combination of genes, and multiple clones of the same will be created so that the desired feature is retained for longer duration of time. And gene transfer is, that is, you're transferring the gene from the bacteria to a plant to get a resistant crop, for example the BT cotton. This type of technology is called as gene transfer. All in all, by biotechnology, we want to concentrate, or we want an aggregation of those DNA segments, those genes which are beneficial, which are having beneficial trait and serve the human mankind better as compared to those which are undesirable. As said before, that during sexual reproduction, the desired genes are aggregated and the undesired genes we always want to get diluted so that we get an organism with the desired features. We have also said that there are various techniques through which we can introduce a gene of interest into the cell so that we can get an organism with a desired trait. Then what does it mean? How would we introduce the DNA into the cell? Or what would happen if we insert an alien DNA into a cell? For example, this is the DNA which is having that gene which is giving the desired feature to a particular cell. The moment I am going to introduce this DNA into the cell, it is not going to divide, it is not going to increase in number because it is not having the capability to divide itself. Moreover, it will be digested by some of the enzymes which is present in the cell. This is N, this is the nucleus which is having the genomic DNA of this particular cell, and this is the alien DNA which we have inserted, and it will not be able to divide itself or increase in its number. So how do we go about it? So what we will do is that we will take this alien DNA and we are going to insert it into a molecule, maybe a chromosome or a carrier molecule or a vehicle-like molecule which is called as vector. Now this particular vehicle-like molecule or the vector is having a sequence of DNA which is called as origin of replication. If the DNA is having this particular sequence, that is origin of replication, it can make multiple copies of itself. So the moment this recombinant molecule which is made up of the DNA and the alien DNA having the origin of replication, when it is inserted into a cell, then since it is having origin of replication, it will form multiple copies of itself. And when the cell is going to divide, even its daughter cell is going to have the copy of this recombinant molecule. This is how an alien DNA is inserted into a cell. A very good example of a vector is a plasmid. A plasmid is basically a circular extra-chromosomal self-replicating DNA molecule. It is circular, it is extra-chromosomal, it is not a part of the chromosomal, and since it is having its own origin of replication, it is capable of self-replication, part of the DNA. It is made up of the DNA, and in this we insert our alien DNA, and the moment we are going to transform this cell, or we are going to insert this recombinant DNA molecule into the cell, it is going to divide and form the multiple copies of itself so that the desired gene is amplified.